Good day everyone, I'm Nobilin Pianita, second year from BSED Science. And now I'm gonna present my final requirements in Bio 023 or Biochemistry. When learning about DNA and RNA replication, nucleotides usually represent one of the first subjects introduced in the topic. The topic assigned to me is the cytosine structure. But before I proceed to discuss cytosine structure, uses, and function, let us have a quick review what is DNA and RNA. The structure of nucleic acids, DNA and RNA. There are two main types of nucleic acids, DNA or deoxyribonucleic acid and RNA ribonucleic acid. Nucleic acids are large molecules made up of smaller molecules called nucleotides. The nucleotides in these molecules are linked together through covalent bonds or bonds where electrons are shared between atoms. Let's start by looking at DNA. The nucleotides that make up DNA have three parts: a phosphate group, a nitrogenous base, and deoxyribose sugar or carbon 5. The phosphate group consists of a phosphate bonded to 4 oxygen atoms with one of the oxygen bonded to the number 5 carbon of the deoxyribose sugar. An important note here, the number 2 carbon of the deoxyribose is bonded to the hydrogen atom. Carbon atom number 1 of the deoxyribose sugar is bonded to the nitrogenous base. There are 4 types of nitrogenous bases that can be found in DNA. Adenine shown here, guanine shown here, cytosine shown here, and the thymine shown here. As you can see, adenine and guanine have double ring structures, and cytosine and thymine have single ring structure. Adenine and guanine are called purine bases. DNA is a double-stranded nucleic acid and its molecules take one helical formation. Each helical chain has its phosphate sugar group towards the outside and the nitrogenous bases of the other chain. Each of the bases of one chain is joined to the base of the other chain through either 2 or 3 hydrogen bonds. Thymine and adenine are joined by 2 hydrogen bonds and cytosine guanine are joined by 3 hydrogen bonds. Thymine and adenine are always a base pair and cytosine and guanine are always a base pair. It's estimated that DNA molecules contain more than 100 million base pairing in one individual's body. The sequence of the base pairing is the same in every DNA molecule. So, this sequence of base pairing is unique to that individual. DNA is often called the information molecule because it contains the master code needed to make various RNA molecules and protein molecules in the body. Now, for RNA, the nucleotides that make up RNA are very similar to those make up DNA. RNA nucleotides have a phosphate group in the same structure as in DNA. They have 5 carbon sugar but number 2 carbon is bonded to a hydroxyl OH group instead of a lone hydrogen atom, and this sugar is called the ribose. It also consists of the 3 same nitrogenous bases as DNA: cytosine, adenine, and guanine. But it does not contain thymine. Instead, it contains uracil, which is single ring structure like thymine, making it a pyrimidine base. Much RNA molecules are single-stranded nucleic acid and many times they form a folded compacted structure with some hydrogen bonding taking place within base pairs of the molecules. These pairings are the same as in DNA. Cytosine and guanine are a base pair and uracil takes the place of thymine and part with adenine. RNA is extremely important in the process of forming different protein in the body. I said earlier it was a quick review pero mukhang napahaba na siya. But, by the way, that is the basic structure of DNA and RNA. And now let us proceed to the main topic, which is the cytosine.
As a noun, a base found in DNA and RNA and derived from pyrimidine. Pairs with guanine.
Cytosine constitute one of the nitrogenous bases including adenine, thymine, guanine, and uracil in forming nucleic acid building blocks, also known by its IUPAC name 4-aminopyridine 2,1-H1. Cytosine is one of the three pyrimidine bases with thymine and uracil constituting the remaining two. Pyrimidine bases are smaller than their purine counterpart due to the presence of a single ring in their structures. By contrast, purine contains two rings. These different in size allow pyrimidine to bond with purines rather, and thus maintain a contained size throughout the double-stranded helices of DNA and RNA. In order, the cytosine must bond with purine counterpart guanine. Aside from its role in creating nucleic acid, cytosine also possesses other significant functions within the cell. Cytosine is a pyrimidine containing a single heterocyclic aromatic ring, a keto group at carbon 2 and an amine group at carbon 4. The molecular is planar in shape. Cytosine can form three hydrogen bonds with guanine. Due to this three hydrogen bond, the cytosine-guanine base pair has an overall higher boiling point and greater bond strength than the adenine-thymine base pair. The high melting point makes the cytosine-guanine base much more resistant to denaturation. The double-stranded of DNA break down into its constituent strand due to the high temperatures.
This is my prelim requirements in biochemistry, the cytosine structure. I use recyclable materials as long as I can. As you can see, the chemical formula of cytosine is C4H5N3O. I labeled each element by color. The black represented the carbon and we have 4 carbon here. 1, 2, 3, and 4. The blue represented the hydrogen and we have 5 hydrogen. 1, 2, 3, 4, and 5. While the yellow represented the nitrogen, we have 3 nitrogen. 1, 2, 3. And lastly, the oxygen. In this structure, we have only 1 oxygen.
The cytosine is one of the 5 nitrogenous bases which make up the genetic code in DNA and RNA. Nucleic acid plays an essential role in heredity, cellular function, and biological reaction. Cytosine can also be methylated by adding a methyl group at carbon 5 position, and in this modified form plays a vital role in epigenetics. Moreover, cytosine can be transformed into other bases such as uracil, further evaluating the importance of this nitrogenous base in the field of epigenetics. Epigenetics is a relatively new field that examines the role of DNA modification in controlling gene expression in organisms. In this epigenetically modified term, cytosine associates with changes in the cellular and developmental process, neuron self-development, and tumor development in human. When bound to the 3-phosphate group, cytosine form the energy-carrying molecule cytidine triphosphate or CTP, which can transfer phosphate group and serve as a cofactor for enzymes. Cytidine forms when cytosine attached to a ribose ring ribofucose via a beta-Ni glycosidic bond. Cytidine represents the nucleoside of cytosine. Unlike a nucleotide which contains a nitrogenous base, a sugar and phosphate group, nucleoside consists only a base and a sugar. In addition to serving as an RNA component, cytidine represents a precursor for uridine also utilized in RNA synthesis. Aside from its role in RNA, Cytidine is also vital for glutamate cycling and glutamate or glutamine levels in brain. Cytosine has a molar mass of 111.10 grams per mole and melting point of 320 to 325 degrees Celsius. It can occur in nucleic acid such as DNA and RNA. It may also be found as component of nucleoside, nucleobase plus sugar, deiribose or ribose, and nucleoside with phosphate group. In DNA and RNA, cytosine matches with guanine, forming three hydrogen bonds. Cytosine is relatively unstable and can be converted into uracil. This alternation can be connected by DNA reverse, such as by the use of the enzyme uracil glycosylase. If not referred, this may lead to a point mutation.